that's a really kind introduction. It also segues into the pitch I'm about to give for students and postdocs who are interested in joining my laboratory. We have lots of interesting projects, and we would love to talk to you about them. So I do want to start by disclosing that I'm going to talk about Oxford Nanopore technology today, and that we do work closely in the lab with Oxford Nanopore. And then I hold stock options. I'm sorry, I'm not a paid consultant anymore for a company called Myome. And then just again, our lab is new. We started July 1st here at the University of Washington, and we're recruiting scientists, students, and postdocs. So if anything we talk about today seems interesting, or you want to talk more about it, I'm always happy to chat with you. And then just kind of learning objectives for today. I think the big picture as we talk about long-read sequencing, understanding the difference between short and long-read sequencing, thinking about when can you use long-read sequencing, when is it the next best test, and I'm going to go through a couple of example cases. And then just ideas about what could it do that I'm not thinking about today. What are some novel applications of this technology? I think it has broad application, and I think there's a lot of exciting things that we can do with it. So I'm going to start with two quick cases. These are two cases that I've seen and sequenced. The first one is just a young child who has prolonged bleeding, and he gets kind of a typical workup that identifies essentially nearly undetectable fibrinogen activity. So the question is, does he have afibrinogenemia or hypofibrinogenemia? So he ends up in a genetics clinic where testing is sent. A bleeding disorder panel is sent. And it identifies a single variant in a gene called FGA. That's the fibrinogen alpha chain gene, so one of those three components of fibrinogen. But there's no second variant found. This is a recessive disease. So my question, and I'm not going to directly answer it just yet, but just think about what clinical test would you send next in this particular case? Like, what's the next best test? Separately, imagine a young boy who has anhidrosis, has joint pain, swelling, dislocations, and has pain insensitivity, who has two older sisters who have essentially identical findings, but also has three siblings that are unaffected. His parents are first cousins, and he gets a clinical diagnosis of hereditary sensory and autonomic neuropathy type 1. Now he has an exome, he has a SNP array, he has an exome reanalysis, and all these are negative. What's your suspicion for a genetic disorder, and then what test would you send next given all these negative evaluation? This is clearly, it's screaming it's genetic, right? But we don't know why. So how would you approach that case? And it's cases like this that got me interested in long-read sequencing, and it's really the workflow that we go through in these cases. So, as I was going through residency and as I was learning how to be a clinical geneticist, the thing that struck me the most is, I don't want to say how bad we are at it, but just how infrequently we actually solve these cases that seem to be genetic. So in more than 50% of cases where we strongly suspect a Mendelian condition or genetic disorder, are we able to actually make a precise molecular diagnosis? And that's after the systematic workup that we go through, which many of you know, right? We do a microarray, it'd be negative, so we then go on to something like a panel or an exome that could be negative. We then might order more specialized testing, methylation studies, karyotype. You pick something out of the clinical, the options that you have. And even at the end of that, we diagnose less than half of the individuals that we suspect have a genetic disorder. So my overall hypothesis, or my overall thought, is that could we use a single technology, long-read sequencing, to both shorten the amount of time it takes to make a genetic diagnosis and also increase the rate of genetic diagnosis. And I hope with some of the data that I'll show you today that you would agree that that's possible and somewhere we can get to in the next few years. So I want to start with just what is long-read sequencing technology. And I find it's typically best to contrast it to short-read sequencing technology. And I know a lot of folks are familiar with this, but just really briefly, short reads essentially are reads that are anywhere from about 100 to 300 base pairs long. They're highly accurate, so greater than 99% per read accuracy. And the cost to sequence a human to about 30x coverage falls kind of every day that you open up Twitter or something like that. So right now, probably less than $500 if you've got the right scale, maybe realistically less than $1,000. The challenge with short reads, though, is that when they align to the genome, they align poorly to complex regions. So this is just a toy example, a region of the genome that's repeated 5 times. With short reads, some reads will align uniquely, right, over the unique and non-unique region. But other reads, they could align to any one of these 4 locations. And you don't know exactly where those will land. And that can be hiding something. Say that there's a repeat expansion in this region, or say that there's a transposable element here that's created a new splice acceptor site, things like that. You can't assay that with short-read sequencing. So long-read sequencing solves this problem by simply being longer than the length of many of the repeats that we study. You're kind of cheating, right? You're just sequencing strands that are longer and they're easier to align or to assemble to the genome. Long reads are typically anything from a KB to greater than 2 or 3 megabases. I think the longest read right now is around, or the longest documented single read is around 4 megabases, 4.5 megabases. The accuracy varies anywhere from 90% to greater than 99%. And the cost, just like short read sequencing, is falling quite dramatically. So now I say it's about $500 to $3,000 to sequence a human to about 30x coverage. But the thing that I want to drive home today is just how information rich these reads are. You can gain a lot of information from the single technology or the single data source. So commercially right now, there are 2 types of long-read sequencing options available, one from PacBio and one from Oxford Nanopore. Illumina is developing a synthetic long-read technology. I've never seen any of the data, and I'm going to try to avoid it. Or I'm going to avoid talking about it today and maybe try to avoid it for the rest of my career as well, because I like the quality of data you get out of these machines. PacBio sequencing works by, this is kind of crazy every time I say it, you're watching a polymerase replicate a circular piece of DNA. So the polymerase is attached to the bottom of a flow cell. It grabs onto circular DNA, and it does what a polymerase wants to do, right? It just adds additional nucleotides. So a camera watches for fluorescence, and when it sees that fluorescence come in, it knows which molecules in the active site of the polymerase. There's 2 modes. One's called CLR and one's called CCS. All I want you to take away from this is that CCS is actually branded as hi-fi sequencing, and these are highly accurate reads, because what you do is you go around that polymerase multiple times, you can zero out, or you can average out, I should say, those errors where you get a really highly accurate read. And these reads are beautiful. The challenges, though, the read length is limited to about 18 to 20 KB, and the accuracy, though, is around 99.9% or so. So really nice data. You can also detect methylation with PacBio. So a methylated CpG, when it goes through that polymerase, will take a little bit longer than an unmethylated CpG. So you can actually detect that change in kinetics to figure out where the methylated molecules are. Nanopore sequencing, just make sure the video works a little bit differently. So what you're doing is you're measuring the change in current as a single-stranded polymer, DNA, RNA, or it could be protein even, but that's a little ways down the road, passes through this protein pore. So these current changes are characteristic, as you can see. So from the current, you can work backwards to figure out which nucleotides were in the pore at any given time. What's nice, there's a couple nice things. The pore doesn't care how long the DNA molecule is. So if you could isolate an entire human chromosome 1, you could sequence it from telomere to telomere if you could get that single molecule through that pore. Also, the pore can pick up any type of change. So however that molecule or the polymer that's in the pore, however it changes that current, you can detect that. So with things like RNA sequencing, so you can directly sequence messenger RNA, and you can also detect the modifications on those mRNAs. There's like 160 or so mRNA modifications. In theory, we could detect all of them using this technology. Right now, we can only do about 6 or so. But folks are working on, and I'm interested in working on, how do we continue, identify more of those? Now the challenge, as many people who've looked at long-read data probably know, is the error rate between the 2 technologies. So on the bottom, I'm showing you PacBio sequencing, data from PacBio, and on the top is data from Oxford Nanopore. And just by looking at this, you can see that there's a lot more noise, even if you don't know what the noise is, in the top set of data than in the bottom set of data. But what I argue is that the essential information is there. You know, this individual is homozygous for this deletion, and you can see that in both data sets. And they're also homozygous for the single nucleotide change, which you can see in both data sets. For me, the big advantage, especially during residency, was the cost and the time to produce the data. It's much cheaper to generate nanopore data, and you can generate much more quickly. Now, just to say that Nanopore has made big strides over time in their error rate. So here I'm showing you data from an older version of the flow cell. And you can see these insertions, these little I's and the deletions are kind of spread all throughout. With a newer version of the flow cell, you see a lot fewer of those. We see improvements in the data with kind of subtle changes in the chemistry and also the design of the pore. The other real fun thing about nanopore is that you can do a lot of interesting things with the pores. And this is an example of something called adaptive sampling. I'm not going to talk about it beyond this, but what I want to point out here is this is real time. So as the DNA molecule is in the pore, you're converting it into A's, G's, C's, and T's. You align it to a genome, and then you decide, does that molecule live in the region of the genome that I care about? If not, you tell that individual pore to reverse the current and kick the molecule out. So it's decided this isn't in a region that it's interested in. Oops, sorry about that, there's a little delay. It reverses the current, you eliminate the molecule, and then you grab a new molecule for sequencing. This lets you enrich for specific regions of the genome during sequencing. This is targeted long-read sequencing. You can do this to enrich. You can use this to deplete. This has been highly useful for a lot of microbial studies, for example. We did this during residency, trying to sequence just specific regions of the genome in individuals with missing variants, for example. So kind of cool things that you can do with this technology with the control that you have over the individual pores. But at a high level, or overall, what I would say is the big advantage of long-read sequencing is the multiple levels of data that you can get from a single data source. So here I'm showing you a screenshot, an IGV screenshot of about 100 or so kb region. This is KCNQ1, so a region that we think about when we think about Beckwith-Wiedemann. So looking at this, it is noisy, but you can call single nucleotide variants, you can call insertion deletion variants, and you could call structural variants as well. You could call it deletions, insertions, et cetera. With the same data set, you can look at methylation. So all I've done is I've changed the view to say, show me the methylation tags. And in this particular region right here, which is over a CPG island, I can see that about half of the reads are methylated, they're red, and the other half are unmethylated, they're blue. I can then go a step further, same data source, single individual, I can phase them into two haplotypes, one of which would be maternal, the other paternal. And you can see the methylation follows the haplotypes as it should for this region. One haplotype's methylated, the other one's unmethylated. And then you can step back and you can look at larger regions. We got good phasing with this individual, even though we had relatively modest read lengths of around 15 or so kb. This is looking at a nearly 1 megabase region. This is the end of chromosome 11. So you have KCNQ1 here, and then you have the two imprinting centers. Somewhere over here. The screen's a little small on the laptop. The point is, though, that you get phasing throughout the region, and then you can look at phased methylation also. So you can see the pattern that you expect. The paternal haplotype is methylated at the two imprinting centers here, so it's red, whereas the maternal haplotype is methylated here at KCNQ1. This is all from a single DNA experiment, right? This is a couple micrograms of DNA sequenced for 3 days and then run through kind of a standard pipeline. And think about how many experiments in the past you had to do to get this level of information. So the first thing I want to just go through is a couple of examples of how I think long-read sequencing could be used to increase the rate of diagnosis. So when you think about why are people unsolved after a standard clinical evaluation, I think one thing that we should always say is that we don't know the function of every gene, of course, right? We need better phenotype-genotype relationships. And I would argue that with long reads, we get additional genotype information. We can see genes that are difficult to sequence using short-read sequencing. But there's also genes where it's really difficult to detect a particular variant or actually interpret that variant. So there are several genes that are really difficult to sequence. So just as an example, when I was a resident on genetics, I saw a kiddo in the NICU, a newborn who got transferred to us for ECMO because she had respiratory failure at birth. We got an exome. There was a likely pathogenic 2 base pair deletion in a gene called Haydn, but only a single variant in that gene. So Haydn is associated with an autosomal recessive ciliopathy. And when you think about newborns who have respiratory failure at birth, a ciliopathy is a good explanation for that, right? The thing, though, was that she only had one variant in the gene Haydn, not two. When we went to look at the gene, we were actually surprised. Haydn's a large gene. It's about 400 or so kb, and it contains a 380 kb segmental duplication. So segmental duplications are regions of the genome that are highly similar to another region of the genome. And these are regions where short reads will align very poorly. So just as an example, if you look at 2 exons from within the segmental duplication, you can see you get poor coverage of short reads to these 2 exons. So you actually couldn't call variation in these regions, right? What if she had a nonsense mutation in one of these exons? You wouldn't see that, right? So our question, and the family's question too, of course, was how do we actually sequence the whole gene, and how do we confirm or exclude a ciliopathy? Because the family was concerned about this. Do they need to see pulmonology long-term? Are there additional screening tests they need to do? So we did long-read sequencing of this kiddo on a research basis, and that gave us really nice even coverage across Haydn. So this is an example of that region that I showed you, where the long-read data, which is shown by the coverage here on top, is smooth across the entire gene within the segmental duplication. And you can see that over the 2 exons with short-read data, we didn't have any visibility. And this isn't a pathogenic change, but it's just a good example. Over this exon right here, she has a heterozygous polymorphism. So she does have some type of variation here that wouldn't be captured with short-read sequencing and could be informative. But in her particular case, we didn't find a second variant. We did confirm this actually with orthogonal, orthogonal study that confirmed our genetic results. The other reason that we might be able to not detect variants is that some structural variants can be really difficult to identify. And it's not really challenging structural variants that are difficult to identify. Sometimes it's pretty simple ones. So that case that I started with, the kiddo with hypofibrinogenemia, who had this single known maternally inherited stop in FGA, we did long-read sequencing of him. We phased the data into maternal-paternal haplotypes, and here I'm showing you one of the two haplotypes. So it's just randomly assigned haplotype 1, haplotype 2. Haplotype 2 here has that known maternally inherited stop in FGA. And the question, of course, is what's the second variant? Well, when you look at the gene, it's pretty obvious. There's a deletion of about 4,000 or so base pairs that takes out the 5' UTR in exon 1 of the gene. But this kiddo had an exome and they didn't see this, which isn't surprising, right? Because if you think about the capture, if it's just looking for that exon and it's trying to say, well, there's 100 reads over exon 2, but there's 60 reads over exon 1, is that a deletion or is that just random sampling? You can't tell that with with high confidence, right? So that's why this would have been missed by standard clinical testing, but was relatively, quote unquote, easy to identify with long-read sequencing. And just as a boot, we get we can phase this as well, which is very helpful. Because with short reads, you may not be able to decide whether or not this deletion is on the same or different haplotype as the maternally inherited stop. And then finally, predicting the impact of a variant can be difficult. There's computational reasons for this. There's lots of reasons for this. So just as an example, this is a kiddo that we saw who, as a toddler, was diagnosed with severe eczema and allergies. And this was the distribution of the eczema in this individual, and standard eczema treatments did not help. And if you've had a kiddo who's had eczema, or if you've, a lot of people have seen eczema, this isn't the distribution that you expect. The family actually went on Facebook and posted a picture, and someone suggested acrodermatitis enteropathica, which is easily treated with zinc, and they gave the kiddo zinc and he got better almost instantly. So the question is, why did he have this disorder? So he came to us, he got genetic testing, and we found a single nonsense variant in the gene SLC39A4. That's a gene associated with this disease. It's a zinc transporter, it made sense. But it's a recessive condition. There was only a single pathogenic variant. There wasn't a second one, but there should have been. So we did long-read sequencing, and this is Excel. Don't judge me for that, but it's kind of a simple way to show kind of the data that we get out. By the time I go up for promotion, I'll change it to something fancier than Excel. But you can see that we have phased haplotypes. So we have haplotype 1 up here and haplotype 2 on the bottom. In the haplotype 1, we see the known pathogenic variant, the stop, that was identified by clinical testing. On the second haplotype, we kind of quickly get a small list of variants that are rare. One's an intronic variant. CAD scores, One's a synonymous variant, probably not doing anything. Splice AI score was not interesting. But there's this upstream gene variant that has a CAD score of 18 that caught my eye. And it's like, well, that's kind of a high CAD score and kind of a, maybe it's something dealing with, it's a regulatory variant. So let's go take a look and see what it does. So we dug into it a little bit, and this was in collaboration with Andrew Stragachis, who's in medical genetics here. This exact nucleotide actually ends up being in a transcription factor binding site that's selectively active during development only in GI cells. So what happens is the transcription factor machinery binds to this, and it keeps that chromatin open, and it keeps it open the rest of your life. With that polymorphism there, the transcription factor machinery doesn't bind, so this gets turned off. And so he's no longer able to express this particular haplotype, or this particular allele, sorry. So even though he has a good copy, he can't read it. So that's explanation for why he has his particular genetic disorder. So we've gone through in these what we call missing variant cases, so cases where we have suspicion for a specific disorder based on prior testing, and we found a variety of variants. These include intronic variants that disrupt splicing. Again, we can phase them into the two haplotypes. These include inversions, which bisect the gene completely, that wasn't identified on karyotype or standard clinical testing. And then also things like transposable element insertion, so like an ALU insertion that hops into an exon. And I really like these missing variant cases because I think they really teach us a lot about, first off, the power of long-read sequencing, but second, what are we missing with standard clinical testing, or what are we at risk of missing? So at this point, I need to update this table. We've sequenced probably about 60 or so missing variant cases. About 2/3 of them we can solve using long-read sequencing. What I think is interesting is the 1/3 we don't solve, right? What are we missing? Even when we have this really impressive data source, what is it we can't see that could be contributing to the disease? And I think those are, each one of those cases is kind of an interesting PhD thesis in a way, right? How can you dig into this and start looking into what's actually the etiology of this disorder? These could be things like rare intronic variants, variants in regulatory regions, and then also variants in other genes that might modify the phenotype, right? Even though it's a recessive disorder, is it a case where you have a variant in the gene associated with it and then also one in a neighboring gene or something like that? So I do think, and we're working on a paper that puts this forward, any unsolved case where you've had a compelling variant in a single gene or a really clear phenotype without a single pathogenic variant, long-read sequencing is probably the next best test, is our argument. So anyone who's got a compelling case, we're happy to take them and take a look at them with long-read sequencing. I want to change gears a little bit and think about how we can use long-read sequencing as a single data source to streamline clinical testing, to make the clinical workflow much simpler and much quicker. This is work that we've done with Kate, who's here in the audience, Anita Beck over at Children's, and then Miranda Gailey, who's the lab manager in my lab. And the question is, how can we use whole genome long-read sequencing as a single test for suspected imprinting disorders? We know that testing for imprinting disorders can be complex. It can be multi-step, so things like Prader-Willi, Angelman, Beckwith-Wiedemann. There could be multiple tests that need to be sent. So the question is, can we use long-read sequencing as a single test for this? What we've done is we've actually developed a tool called Meow. This was a name and a logo in search of a use, so that's how we ended up with this. This was actually written by Miranda, who again is the lab manager in my lab. So we know that there's about 31,000 CpG islands in the genome, and the question is, can we actually evaluate all of those CpG islands simultaneously, and then identify relevant differences in methylation that could point us toward a particular disorder? So let me show you an example of that. So this is an individual, a control case of Prader-Willi syndrome. This is coverage that's been plotted, and then the different colors are the different genes along the chromosome there. And you can see that with the coverage, it kind of trucks along here and then it drops off. And this represents a deletion right here. So using the same data source, using long-read sequencing, you can get information about the polymorphisms as well. So we have homozygous polymorphisms here on top and heterozygous polymorphisms on the bottom. So you would expect within a deletion that you would lose all of your heterozygous polymorphisms, which is what you see. So it's a nice confirmation that you've actually got a real deletion here. You can then go a step further and you can add that methylation information in from those individual CpG islands. These are select CpG islands. There's a lot of CpG islands throughout here. But the one that I want you to focus on is this particular one right here that's in SNRF and SNRPN. So there are 3 CpG islands, but when you have Prader-Willi syndrome, there's actually one specific one that ends up being hypermethylated. And Meow will pick these out individually throughout the entire genome. So we compare these to our control database that we have. And one thing we need to do is expand our control database. But so far in the cases that we've looked at, and we've been blinded to all of them, except for a couple, we're correctly identifying the actual aberration that was known, just using either methylation by itself. And this is the exact CpG that's tested clinically as well. But of course, one question that we have and that a lot of other people have as well is, what if you don't know exactly where to look when you're doing this testing? And that's a question that I put to Miranda. I gave her a case where, I knew the disorder, but the question was, what do you find if you just look using methylation? So she ran the tool and found 3 differentially methylated CpG sites. One at SNRF-SNRPN, where there was hypomethylation, so the individual had essentially no methylation, whereas most individuals have about 50%. And then 2 other sites where you get some hypomethylation as well. So going and looking at this particular site, we see that it actually, of course, falls within the critical region for Prader-Willi or Angelman syndrome. And there's a large deletion of this individual as well. So this is a case of Angelman syndrome, but this was identified just by looking at the CpG islands, right? Not by looking at coverage, not by looking at SNPs, anything like that. So it teaches us that we can actually use methylation information to go back and look for well-known disorders. We also see some interesting age-specific differentially methylated regions when we look. We have a project where we're sequencing trios from the Seattle Children's NICU using long-read sequencing and comparing them to the short-read sequencing result. This is just one example from that project where we have a trio. We have mom, dad, and then the neonate. This was an unsolved case. We ran Meow, and we asked, where are the differentially methylated sites? So within this gene, there's 2 CpG islands here. And you can see that this particular CpG, they're completely methylated. But at this CpG, mom and dad are both unmethylated, but the proband is 50%. And when you phase this, I'm not showing it, but when you phase this, that methylation is assigned to a single haplotype. So it's one haplotype that's methylated, the other one that's not. And this was flagged by Meow, so it's something that caught our eye. And we thought, oh, this is interesting. Maybe this is, could help explain the phenotype in this individual. But then we said, these are newborns. Maybe we should compare them to other newborns. And actually what ends up happening is that for age match controls, we do see this CpG island that's about 50% methylated in other neonates. So while it's an interesting hit, it teaches us that there's these age-specific effects that we need to be aware of. But it's not just that you can use this as a way to filter out things, but it's also kind of interesting physiology, right? Like why is this 50% methylated in some individuals and not? So it kind of can drive us down this path of what's actually going on. Why is this here? I want to switch, again, just to think about how could we use long-read sequencing to get to a diagnosis quicker, and how quick could we actually get to that diagnosis? So at the beginning of 2022, end of 2021, a nice paper came out in the New England Journal from you and Ashley's group at Stanford, where they used Nanopore in the critical setting, or in the critical care setting, to sequence, I think it was 12 individuals who had suspected genetic disorders. These were adults and kiddos. And what they did is they basically timed themselves. They timed themselves from when they did their DNA extraction to when they actually got either confirmed, or they came to a reasonable genetic diagnosis, or said we can't figure out exactly what this person has. And the fastest time for them to go from blood to diagnosis was around 8 hours or so. And they make a big deal out of this, and they should have. It was an impressive accomplishment. They got a world record, and there were a lot of balloons involved and things like that. So, our question was, given this workflow, and this is something that we had talked about doing for quite a while, we thought, could we actually do this, first off, quicker, but also, second, using less blood? Because in the populations that we're thinking about, in neonates, you don't really want to start with 3 to 5 mils of blood. You want to start with less, because there's daily blood draw limits, things like that. So we had a family who was pregnant. So they had a child who had a recessive Mendelian condition. They were pregnant with the genetic sibling of that child. These were adopted embryos. And what they wanted to know was, quickly after birth, is this new child affected by this genetic disorder? And they wanted to know this because the disorder the kiddo had responded really well to zinc. This is the example of the zinc deficiency that I showed you earlier. The challenge with getting prenatal testing was that the second variant we found, that promoter variant, was a research variant, right? So they couldn't do clinical testing using that. So the question simply was, how quickly after birth could we figure out whether or not this kiddo inherited those 2 risk alleles? So this is work that was really led by Miranda, again, the lab manager in my lab, and then Pax Reed, who's a research scientist in the lab. And we developed a protocol that basically said, how quickly can we go from blood to result? And I'm going to kind of walk through this in steps just to highlight the points. So kiddo was born around 10:30 at night here on the 5th floor, and we got about 2 mils of cord blood. And it takes, if you're curious, 7 minutes to walk really, really quickly from labor and delivery over to the I wing, up and down some stairs. It took us 37 minutes to isolate DNA, which it was me doing it, so it was slower than it should have been. It takes Miranda about 20 minutes to do it. This was a column-based approach, and we did a column-based approach on purpose because we wanted something that could be automated more easily in the clinical laboratory. You can do a really nice cell-based approach, something like a Monarch kit, but we didn't think this would be something that would be easily tractable in the clinical lab, so we wanted to do something that was more real world. We then just quantified the DNA just to make sure that when we do our sequencing or when we make our libraries that we actually have quality DNA. So in total, this process takes about 45 minutes or so, but I really think it could be cut down if it's not me doing the DNA isolation. We then prepared the DNA libraries for sequencing. We use a special kind of library kit called the Rapid Kit from Nanopore. This is a transposase-based kit, so basically you add the transposase to the DNA, it randomly integrates, and that creates a new site for those library adapters to attach to. In reality, it takes about 10 to 15 minutes to do. Because we were making a lot of libraries, it took us about 25 total minutes to make these libraries for sequencing. We then loaded the flow cells for sequencing. We did 20 Promethean flow cells. So just to give you an idea of the scale, a single Promethean flow cell can sequence a human to about 30 to 40x coverage over a period of about 3 days. because we wanted to move rapidly, we used a lot of flow cells to generate a lot of data at one time, and then we would wash and store those flow cells. So we prepared the 20 flow cells. So as I was making the libraries, Pax was getting the flow cells ready for sequencing. And then once the library was ready, Pax and I went through basically shoulder to shoulder loading the flow cells. That's not really tractable in the clinical laboratory, right? We need a better way to do that. But we need to figure out what we can do and where the challenges are before we can improve it, of course. And then we started sequencing. Now, the nice thing is, as I mentioned with adaptive sampling, that sequencing data is transmitted to a computer in real time. So as the signal comes off of the sequencer, we synced it to a base calling server, which then in real time base called all the data, aligned it to a genome, and then just kind of constantly gave us a new aligned genome file. So the question is, over what period of time could you actually, or how long would it take you to actually get to be able to call the known variants? And it took us 2 hours and 47 minutes from when the child was born to identify that he did not inherit those variants that his genetic sibling had. In the paper that we wrote, we say within 3 hours because when we wrote the code, we didn't expect to get enough data within 3 hours. So the first time the base caller kicked off was at 3 hours because we had it all set up so it would just go. But we knew at about 2 hours and 45 minutes that he didn't inherit those variants. So these are known risk alleles that we were evaluating. This wasn't a de novo, going from nowhere, but you can imagine that the process would be essentially the same. So I kind of want to wrap up just by thinking about how will long-read sequencing impact all these different areas of medicine. And I'm really, I'm optimistic about this. I'm excited about this because I think there's a lot of projects that will benefit from long-read sequencing and a lot of different assays that could benefit from long-read sequencing as well. And the first is simply prenatal testing. With cell-free DNA, we can isolate cell-free DNA, of course, and sequence it on a nanopore machine or on a PacBio machine as well. And given how the technology works, especially nanopore, you can essentially get that data in real time, and you can imagine shorter turnaround times. You don't need to get 10 or 20 samples. You can do one sample at a time, as they come in. That gives you a quicker turnaround time, which can, it can matter based on how laws are changing, honestly, in the United States, right? You want to know things sooner, and this kind of technology can give you that. It also gives you the ability to do real-time analysis. You can imagine that as the data is coming in, you can see kind of, is there a deletion? Is there an extra chromosome? Things like that. You also get methylation along with the nucleosome position, right? So the fragment, the position tells us about the nucleosome, but we also get the methylation as well, which can give us more confidence when we're separating that maternal and fetal reads. Next, I think a lot about how we could use this for cancer testing. First, again, shorter turnaround times. I imagine a day where an individual comes into clinic for a visit, they get blood drawn in the morning, we sequence their genome in 1 or 2 hours, and then we have a phased assembly ready for their clinic visit in the afternoon. That gives us a more comprehensive view of the tumor. It gives us methylation as well, right? So we can use that maybe to predict things like recurrence or response. And then finally, one area that I'm especially excited about that we're just starting to think about how to develop is how can we use this for rapid pathogen detection? As you sequence these, you get that information in real time. So do you get quicker turnaround times? Do you get more comprehensive view of the pathogens that you're sequencing? Could you sequence a resistance gene, for example, at the same time? And then you can think about things like handheld devices that might end up in the emergency room. So this is a mock-up from Nanopore. This is a sequencer that I've only ever seen made out of plastic. I've never seen it work. But the idea is that it attaches to a device like a handheld phone. If you could come up with a way to take blood, get it through a library prep, some type of microfluidic device, and then get it loaded on the flow cell and sequenced, you can imagine where you have your sequencer going at the same time your other POC labs are going, like your lights or your glucose, things like that. I don't think this is too far-fetched, and I think we have kind of the pieces in place to make this work once we can actually get the practical piece of getting the DNA out of blood, getting it sequenced, things like that going. So I do think the future is really bright for long-read based, for long-read sequencing in general, and then I think more specifically for long-read sequencing based clinical tests. I'm going to watch Kate while I say some of this to make sure she doesn't throw anything at me. What my hope is that within the next couple of years, we have a long-read sequencing based test that can be used to just phase known variants. This happens to us a lot in genetics clinic. We get an individual who has a de novo variant in a gene associated with the disease. We can't do parental testing to figure out segregation. So if you do long read, you can see, are they on the same or different haplotypes, right? The other is clarifying structural variants. So you do a microarray, you see a duplication, and the question becomes, is it in tandem or not? We used to do mate pair testing at Mayo. That's not available anymore, so there's not a good way to do that clinically. Long-read sequencing is a really straightforward way to do that. And then again, as I mentioned, a single test for imprinting disorders. We suspect Prader-Willi, we suspect Angelman. Could we use this as a single test for that? Within the next 2 to 5 years, think about cases where we have missing variants. We have an individual with PKU, what single variant PAH, for example. Can you use this to look for that second variant? Doing single gene deletion, duplication testing, and sequencing all in one go. You just do whole genome sequencing, and then you analyze that specific region that you're interested in. You can also think about things like rapid aneuploidy detection for products of conception. We can do that for, if you're in MFM clinic, you do an ultrasound and you see something abnormal, can you, get mom's DNA or get blood and do cell-free DNA testing quickly? Is that possible using this technology? And then, of course, kind of more long-term, a little bit more pie in the sky, thinking about using long reads to do variant calling from phased assemblies. That will simplify a lot of these challenges we have with identifying things like that deletion that I showed you in that kiddo with hypofibrinogenemia. Ultra-rapid testing, of course, like how quickly can we turn these around? Can we get to the point where a team admits someone in the morning and they get the genetic testing results by the afternoon? I think with this technology, we can get to that point. And then you can start thinking of, I don't know, maybe I put this in here and maybe it's a little bit crazy, but that's fine. Say you install a device at a satellite location, you do that sequencing on the device, but you transfer that data back and you do the analysis yourself. This is the kind of technology I think that makes it easy, right? Because the library prep can be simplified. You can think about microfluidic type devices that just take the blood and then run the sequencing for you. It can really help with logistics, right? How do you collect that blood? How do you transport it? Things like that. So things can be done a lot more quickly. So I'll stop there. I kept it a little short on purpose because I hope we'll have a little bit of discussion. I want to say first, thank you all for the opportunity to present today. I always enjoy talking about long-read sequencing, and especially around a group of folks who I think kind of think similar to me. Of course, I want to thank the BBI for the funding and NIH as well. We've had lots of collaborators at Children's and at the University of Washington. If I had to list all the collaborators, it would just be slides and slides and slides. So I just say I've worked with a lot of people here, and I appreciate all of that. And then just as a last plug for students and postdocs, we are hiring, recruiting, and would love to have the opportunity to chat with you about what we do. So thank you. Are there Zoom questions that we should field, or are they? Not yet. Not yet? I think I'm supposed to show that next.
I have a question first. So the quality, sequencing quality, and the cost, you're talking about these trade-offs. I just wonder, what's the barrier for what's the highest cost piece? And also, what's the, are there ways that that bioinformatics can improve the quality? Like, for example, if you have a long read, does phasing different variants help you identify kind of, this is real because it's in the same phase the other one you've seen?
Yeah, so Brian's question is a good one. It's, how can you improve your quality based on the different technologies? And does having more quality or less quality, how does that affect your variant interpretation? So in no particular order, something like phased assemblies, it's much easier to do things like structural variant calling, right? Especially if it's complex, multiple rearrangements. If you can assemble that and then do some kind of, look at the graph, right, and figure out what the path through is, it gives you a much clearer picture than if you're trying to call based on how the aligner align the reads to the genome. The differences in quality, so again, PacBio, it's much more expensive. Right now, to get a 2030X genome, materials is probably around $5,000–6,000. That's going to drop with the new Revio machine that folks you might have seen that was announced at ASHG. They claim $1,000. That doesn't include the cost of the instrument, so I think the real cost will probably be about $2,000. And then that nanopore cost continues to fall and is probably around $750 or so right now. There will be some improvements to nanopore that are announced soon next week at their community meeting, which will probably increase that quality more. But we always have to be thinking about coverage, right? So like, if you had a perfect read, but you only had a single read, would you trust a pathogenic variant in that single read? You probably want 4 or 5 of them, right? So at some point, it's not, you just don't need more quality, it's just good coverage of the region as well. So I think lots to consider with the two technologies. I have an example of a case that we missed because of the error rate of nanopore. So things that we need to keep in mind when we look at this data as well. Yep.
Thanks, that was a wonderful talk. I had a question on the ability of this technology to sort of solve the missing variant where you find one but don't find the other. So in a case like you presented, where someone, a child, and this, I'm asking for your intuition sort of as a clinical geneticist, if you found a clinical picture that looks like eczema that responds to zinc, and the family treats it with zinc and it goes away, and then you find the causative gene, you find one of them. In Bayesian terms, are you already there? Because I'm thinking about trying to convince someone to pay you to do that second, to find that second one.
You're probably there, right? Like we all agree on that. Yeah. Say they want to have another child, what's their risk? How can you appropriately counsel for that, right? Say they want to test against that. How do you do that? Do you just test it against the single variant? So I think there's ways to say this is why we should do that additional test. So that maybe more practical piece aside, scientifically, say you look and you don't find a second variant, but they have the disease. So what are we missing? What's that pathway? What's that mechanism that we don't understand? And what might that lead us to, right? Could it be an enhancer that has a variant that's then causing the second allele not to be expressed? Or is it 20% expression isn't enough? Things like that. So, getting people to pay for it's one thing, but then it's this is really interesting, what can it teach us piece as well, I think is the important piece. So, but yeah, it's a good practical question.
So, Dan, you've worked on a wide number of cases. Is there one that stands out to you as maybe most surprising in terms of what you found in Longread?
I think about the inversion. Kate will remember. Young adult with Lesch-Nyhan, biochemically confirmed, never got a molecular diagnosis, had a karyotype that was negative or normal, I should say, and then Shortread targeted testing on an exome backbone was non-diagnostic. So we did long-read sequencing and found a 17 megabase inversion that bisected HPRT1. I went to Kate and I said, here's what we found. So Kate said, well, would we have seen it on the karyotype? So we pulled up the karyotype and we stared at it for I don't know how long, and we're like, maybe, no. And Kate designed FISH probes that proved that there was indeed the inversion there. The important piece of that case to me is that he had a sister who wanted to know what her risk was of having a son with Lesch-Nyhan, and no one could ever tell her that. So now we had a relatively easy way, FISH, to figure out if it was inherited, and then if she had that as well, so she would know what her risk was. So, and that was one of the first ones that we ever solved, right? So it kind of proved to us that, like, hey, this is going to be good, right? This is going to work well. So, yeah. Oh, sorry, Paul. Yeah.
So you mentioned a possible application of the sequencing to non-invasive prenatal testing,
Yep.
and you said one possible advantage is the shorter turnaround time. For standard non-invasive prenatal testing, of course, one of the limitations is low fetal fractions. Do you anticipate the sequencing you being able to use it earlier in pregnancy?
That's an excellent question. So how early could you use long reads in IPT, and does the fetal fraction affect that? I have to be honest, I don't know, but that's one question that we have as we're kind of thinking about how to design those experiments. Like, what does it look like at 9, 12? Pick your weeks, right? And then ask, what can we actually see? Could we go earlier if we get things like methylation, right? Is there a methylation signal that we can pick up and get a better sense of what that fraction is? I don't know, but it's going to be part of that evaluation, and it's an excellent question. Yeah.
Yeah. So does adaptive sampling work well for cDNA sequencing? Is that something that you could use for some unresolved cases?
That's a question a lot of folks ask. So does adaptive sampling work for cDNA, or can I take it a step further and say for direct RNA also, right? So could you enrich for rare transcripts or get rid of stuff? The problem is it takes about 500 base pairs before you decide whether or not to make that reject decision. By the time you've decided, you're with a typical RNA molecule, you're probably most of the way through. So you should probably just keep going, right? If it's only a 1 kb molecule, it's going to take you that much time to reject it. So just go ahead and run it through. Because adaptive sampling is a little bit harder on the pore.
Yeah,
so you get shorter flow cell lifetime, or it doesn't last as long, right? So then you get less data. So you should just go ahead and blow through them all, right? I think what we'll see for things like cDNA is we'll up the speed of the motor protein, so send the molecules through faster, which will give us a better sense of those rarer transcripts. But
This is Colin asking, it's a great talk. Have long-read technology been applied to cell-free DNA? I talked about, with maybe other forms of cell-free DNA,
Yep.
Which is by definition short reads.
Yep.
And specifically, that one asked about the methylation. You may have already answered this.
Yeah, so it's there's a lot of papers that have come out recently about cell-free DNA in cancer using that predict recurrence risk, and then also, can you see a different methylation pattern that could clue you into something going on? So it's a good question. There's a lot of recent literature, and I'm happy to share that offline. But it is certainly something that we're interested in looking at as well. Like, how could we evaluate individuals and try to predict that, predict it better using things like nucleosome position or methylation? Yeah, because otherwise you just do short read sequencing and you get most of the molecules, right? But you do see longer molecules. There's a paper that came out in PNAS a few weeks ago, probably now, or a few months ago, I'm sorry, where they report, getting 3, 4, 5 kb molecules not infrequently in cell-free DNA. So what information can you gather from those? Probably something, right?
I had a question, not cell-free DNA. But on the quantitative methylation side of things,
Yep.
I'm wondering about the reproducibility. So if you think about on the germline side, it would be maybe Becker-Wiedemann syndrome, other syndromes. But on the cancer side of things, right, we're doing diagnostic, prognostic when you're thinking about quantitative methylation. What is the reproducibility like?
Yeah, what is the reproducibility in cell-free DNA for methylation? We don't know yet, right? We need to do those studies to figure that. We're all in new territory here with this, right? Which is partly exciting, but also we have to ask those most basic questions, right? This could be a bust. I'm not naive, right? But I think it's something that we need to look into because I think there's a lot of promise there as well. So yeah, excellent question. Yeah.
Another question for you, kind of building off of the questions you've been getting. I think I heard you say at one point it took you 3 days to do some of the sequencing runs. Is that true?
Yeah, the flow cells typically quote unquote live, or they're good for about 72 hours. So then you can you can actually choose to sequence for like 5 hours, wash, store them, and then come back and use them again as well.
So as you start thinking about methylation, RNA sequencing, RNA modifications, some of this stuff ends up being much less stable than traditional DNA and nucleic acid strands.
Right.
Is anybody talking about stability over 72 hours?
I haven't seen it, but that's a good question. Like, how does your profile change over time? I've detected more of modification A within the first 3 hours. I don't see it after 6
Right.
hours, right? It's an important point that I haven't seen talked about yet. Yeah, I know. This is all new. We're all learning. Brian?
Someone else on the line's asking, are there situations for which long-range sequencing will not work well and for which other sequencing approaches would be better?
Yeah.
Not counting cost.
Somatic mutations are difficult to detect because you need more coverage, right? And the error rate with nanopore. With PacBio, it's going to be better at detecting those, but if you need 1000x coverage, long reads probably is not the right way to go until the cost comes way down. And then other cases, you spend so much time thinking about one thing, it's hard to see the disadvantages, but I guess the somatic mosaicism is the biggest one that I think about for this. Yeah, Anna?
I guess what about known familial testing? Like if you know what the variant is already, was that more cost effective and faster through traditional short read versus long read? Or do you still think there may be layers of information there?
So is it, if you know the variants, if you're doing familial testing, is it more cost effective to do short read? That piece is a bit, magic to me. So I would actually have to defer. I don't know. I think it's a fair question to ask. Like, does it make more sense to do long read in those cases. You know, in cases where you have to phase, and technically it won't work with short reads, of course you'd want to use long reads, but you know, it could be more cost effective using certain technologies, yeah. Okay, and with that, I will say thank you everyone again.